Okay, so it's recording. So first of all, welcome to our first webinar, and thanks for joining us. This is the first webinar that we are offering, and then hopefully if there's interest in continuing with this from you guys, we'll have a webinar every month, and we'll announce the topics on the website, and we'll post on our social media channels as well. So for today, I'll be talking about the basics of honeybee biology. So my name is Renata Borba, and I'm the Tech Transfer Program lead here in Alberta. I started as the Tech Transfer Program lead last year, and I have been working with bees for quite a few years. When I was young, I used to tell my parents that I knew for sure I wanted to work with animals. I knew I wanted to work with animals, and my mom would ask me what kind of animal I wanted to work with, and I would always say, I'm not sure, but I want to work with a big animal. Like, I want big animals. And here I am working with bees, and I have been working with bees for about 14 years, I think. So have been working with bees for quite some time, and I quite enjoy and I love bees. Before we start with the biology slides, I did receive a question. So for this webinar, I would like to have, before we have a presentation, I'd like to have a bit of a chat with you guys and maybe answer some questions that you may have. So I ask for people to email me questions if they have any questions, and I will try to answer. But when you email me beforehand, that gives me a chance also to do some research if I don't know the answer, right? So that's a good idea. So I did receive a question about deformed wing virus and a nice photo. I circled here the bee in question that has the deformed wings, and that's a sign of deformed wing virus. That's what we see when a bee is showing signs of that disease. And deformed wing virus is a virus that is transmitted by mites, by varroa mites. And the person asked me to talk a little bit about deformed wing virus and maybe explain about the disease. Well, it's transmitted by mites, and as other viruses. The problem with deformed wing viruses and other viruses transmitted by mites is that many times when you see that, it can be too late. So if you do not control your mite levels, by the time you see those signs, it can be too late, and the colony already has a high level of that virus. So you can control your mites, but by that time, it has already established in the colony. High mite levels, it is an indication that, so when you have high mite levels, we do say, well, it's kind of likely you have high viral levels as well. But many times you can have low mite levels and still have high viral levels, deformed wing virus, for example, because you're seeing something after the fact already. So you controlled your mites, okay, but the viruses are already high. And by that point, there's really nothing you can do because we don't have anything to treat viruses. So the best way to avoid having high viral levels is controlling your mites from the beginning, not let it get high. So I think that's the short answer. There's a lot to say about viruses, and maybe we can do a webinar another day on pathogens and viruses and things like that. So to start with our presentation today on bee biology. The first question I think you need to ask yourself is, do you want to be a better beekeeper? And if you are watching this, if you are here today and you want to learn about bee biology, I do think you want to be a better beekeeper, because we need to understand about the biology and about more about the animal, the organism, the insect that we are working with if we want to succeed in that. So if you want to be a good beekeeper, it is very important to learn about the biology and the behavior of bees, but also the pests and the pathogens of honeybees. Because if you know about these, then you can then have a better idea on how to treat and on how to fix a specific problem, for example. So the first question, and that's where the name come from, is honeybees. So why do bees make honey? Well, bees, they obtain most of the energy from carbohydrates, and these are sugars. Carbohydrates are sugars, and that's what nectar is. Nectar is mainly sugar, and that's the carbohydrate source for bees. Sucrose, fructose, and glucose are the most common sugars in nectar. And it's important to mention that some sugars can be toxic to bees. An example of a sugar that is toxic to bees is galactose, and that's toxic to bees. Bees, they break down sugars in nectar by adding enzymes, and they dehydrate the nectar. So at that point, we call it honey. And when they dehydrate the nectar and they sort of process the nectar into honey, they do that to preserve the honey, to preserve that food. Because bees, they do not eat that nectar that they collect right away. They store it for a long term, and they needed to find a way to store it for a long term of time without that food going bad, right? So that's what honey is. It's a sort of processed food, but it's processed by the bees by adding enzymes to it. And the goal is to create a food for them that they can store for a long period of time without going bad. And we all kind of know, there's the saying that honey never goes bad. And that's exactly why, because bees need that honey for a long time, and they need to have something that doesn't go bad. Bees need a lot of food storage for the winter, and they can't afford, so they can't have any fresh nectar. So they need that food to be preserved. Other bees, for example, bumblebees, they do not add these enzymes. They do not make what we call honey. They do not have honey in their colony. They have just basically nectar. As you can see here, pointing by this arrow, is the pots. We call the honey pots, but it's just basically nectar. Bumblebees don't need to store food for a long period of time. They collect the nectar and they utilize that quite quickly, so they don't need something to stay good for a long period of time. So honeybees are different in that way, because they do their colony live for many, many years, and they do need that kind of food. Bees collect also pollen, as we all know, and I will talk a little bit about pollen and how they use pollen, what pollen is good for in their diet, their nutrition, what is it for. But pollen is their protein source. It's their main protein source, and honey is their carbohydrates source. Bees also collect water, and this is very important, because a lot of people don't know that, or don't really give water the importance that it is for bees. And it's very important that bees have a source of water near their apiary. So if there's no natural source of water near your apiary, it is very important that you have something in that yard for them to collect water. Bees, and I will talk about this a little bit further, but water is very important in the summer for the bees, and also early spring. And bees collect resin as well. Resin is a sticky exudate that contains antimicrobial compounds. It's mainly in trees, but some shrubs also produce resin. But it's a product of the trees. And trees produce that as their own defense mechanism. They use resin to coat young leaves, as you can see on the image on the corner there. So when they have young leaf buds, they will coat the entire leaf bud with resin to protect that young leaf from herbivores and also pathogens. And they will also induce a defense mechanism by producing resin in specific places when they need to, the tree. So it's a defense mechanism of the plant, but many insects use, and even mammals use resin for their own benefit as a medicine. So, and that's the case for bees as well. Bees collect these very rich antimicrobial compound from trees, that it's resin. And when they bring it to the colony, we call it propolis. Mainly the difference, the word here is that resin is a tree product, and it's very specific to each tree. But propolis is a mixture of different resins. And when they bring to the colony, they also end up adding some mandibular secretion. They collect that with their mouth parts. We don't know if they actually intend to add these secretions to resin, or if it happens just because the way they manipulate the resin. But we do find in resin some mandibular secretions as well. So in propolis, we found in propolis mandibular secretions as well. And they also add wax to it. But that's basically what propolis is. And you can see on that behind leg, the green ball there, that's a ball of resin. And they collect from trees using their mouth parts. They bring back to the hive in their hind leg, the pollen baskets. And because it's so sticky, they need the help of another nestmate to remove that resin ball from their hind legs. Many times, just so you have an idea, pollen unloading, when they come with pollen from the field, pollen unloading task can take even like 1 to 3 minutes. So it's very, very quick. But resin unloading can take up to 30 minutes just because it's very sticky. So here on the bottom image, you're seeing a house bee assisting with the task and removing that resin from their hind leg. It's quite neat to watch. They're doing that. So after the house bee removes that resin from the hind leg, it will deposit in the hive where that propolis is needed. So bees usually deposit on top of the frames. They will deposit in cracks that the hive has, and when the frames touch, that creates a crack, and they will deposit propolis there. And they also coat the inside of the colony with propolis. And when they do that, we call it the propolis envelope. So here in this photo, this photo is from a nest inside of a tree. So these bees were nesting in their natural habitat, where the surface is very rough. So it's inside of a tree. It's not the same as in a colony, the man-made boxes that we use today, because those boxes are very smooth. And what we think is that those smooth surface does not trigger propolis deposition because it's smooth. But inside of a tree with very rough surface, we do see that the bees will deposit propolis surrounding the nest and making what we call that propolis envelope. I did a lot of work studying propolis and the benefit of propolis to bees, and we found that that external antimicrobial layer, because that's what that is, that propolis has antimicrobial properties. So we found that that antimicrobial layer promotes not just adult bees, but also larvae antimicrobial defenses, and helps them basically to stay healthy, and also reduces bacterial and fungal infection. This has been studied by not just by myself, but by many other people, and the results are quite consistent, and we believe that the presence of a propolis in the colony is very important for the colony, and is a critical structure, let's say, to that colony, to have that external antimicrobial layer. Unfortunately, we don't see bees depositing that propolis envelope in regular colonies, in man-made boxes, because of the smooth surface. When that surface is modified to create a rough pattern to resemble a tree cavity, then the bees deposit propolis. So it's quite interesting. Now, bees are very organized. They like their nest very, very organized. And if you have your own bees, you have noticed this. They keep the nest on the bottom. So the queen and the brood is usually on the first or first two boxes, depending how many boxes you allow them to grow their nest into. Some beekeepers run in single brood chambers, others double brood chambers, but they keep their nest below the first or second box, and the honey on top of the nest. And that's quite convenient for beekeepers, right? Because if they need more space, or if you want and they're right on top, so you don't have to go too much into the brood nest and just disturb the colony. So bees are very organized in that way. They also have what we call the division of labor. What that means is that bees from different age will perform different tasks. With bees, as they age, as they get older, they will change their tasks, and that in some way, it kind of, that the task that they will do kind of grows into them. So some bees are nectar foragers, for example, and they will mostly only collect nectar. They're very specialized. And some bees are pollen foragers, and they will rarely switch to nectar foraging. So it kind of grows into them. There's a few hypotheses on how that happens, but today I will just kind of touch on all this. But yes, they're kind of specialized, and as they age, their tasks change. They have some flexibility there. So for example, if something happens and you lose a cohort of age bees, if your hive goes queenless, for example, that will happen. You will lose a cohort of age, some at a specific age at some period. But bees are a little bit flexible. So when that happens, they can sense that they're missing house bees, let's say. So bees that are a little bit older and have passed that task already, they can reverse back and start doing the task of younger bees. And the same goes for older bees. So if you lose that cohort of older bees, younger bees will start foraging early, so they can forage and bring more food to the colony. So they're a little bit flexible. It does not mean that they will only do that task at that specific period of time and that's it. No, there's some flexibility there. So this is just a graph with some age range, so you can have an idea. But basically, what we're seeing here is that younger bees, they tend to stay inside of the colony. So younger bees will do inside tasks, and that includes cell cleaning, capping brood, tending brood. They will also feed the queen. They will receive nectar, handling pollen, and build comb, and do all that. So younger bees will tend to stay inside of the colony, and as they age, they start to perform outside tasks, and that will include ventilating or protecting the colony. So they're the ones that stay by the entrance, and the last one is usually foraging. They'll go out and forage. Worker bee lifespan is about 3 to 6 weeks in the summer. So those are I don't like to call summer bees because it also includes spring and early fall bees. I prefer to say non-winter bees. Lifespan is from 3 to 6 weeks. Winter bees are much different physiologically as well. So they live much longer. They can live throughout the winter. So those are different bees physiologically as well. But in general, the non-winter bees lifespan would be 3 to 6 weeks. So bees go out and forage, and when they come back, they will dance to communicate resources. And there's two main dances that bees do. One, it's very famous, is the waggle dance, and most people know about that one. But the waggle dance, the forager that went outside and found this nice patch of canola, comes back and wants to tell all other bees and recruit more bees to that patch of nice canola field, right? So he comes back, and basically the waggle dance is for longer distance fields or resource patch, not for the short distance. And the longer distance dance, the waggle dance, they can tell the recruiters, the other foragers, how far it is in the direction of that resource based on the sun from the colony. So it will tell the direction, and we will tell how far it is. But bees do not communicate distance like us. So it's not like they're saying, hey, that nice canola field is 3 kilometers from here. If you go northeast, no, it's not like that. Bees communicate basically the distances, how much energy you will need to get to that patch and come back. So if it's windy, it will take them more energy to get there because it's windy, and in either way they will be getting a headwind, and they will have to fly a little bit harder to get back or to get there. So if it's windy, they will say that it's further away, just because they would need more energy to get there. They also give a sample of the resource that they are communicating about. So the bees can have a taste for it and I guess decide on their own if it's worth the time or not. The nearby distance dance is called the round dance. And those is for very close by floral resources that the bees is communicating to. For that one, the bee is not telling the direction. It just tells maybe how far, it just tells how far it is, but it doesn't tell the direction. It doesn't tell them the direction according to the sun from the hive where that patch is. And that's basically because what the bee is saying is like, get out, you can't miss it, he's right here. So they don't tell the direction, but they do tell how far it is, and they give also a sample of the resource that they're dancing. How much honey do bees need? This is a question a lot of people ask, and they also ask, how can the bees survive if we're stealing all their honey? Well, the thing is that bees are kind of hoarders. They will collect a lot of honey, and they're not good at math, so they would just collect as much as they can. They're not keeping track. So as long as you add supers, as long as they have space, and of course there are resources for them to collect, they will store honey. So because they do that, because they would just collect more and more and more honey, it is up to you, the beekeeper, to figure out how much honey your colony needs to survive the winter. Okay, so the bees will not do that for themselves. You have to figure out how much honey your colony needs, and you have to take out only the surplus so that colony can survive the winter. Just for reference, if you have read the Beekeeping in Western Canada book, one of the guidelines for a double brood chamber colony is that they have between 70 to 75 kilograms of the weight, the colony, 70 to 75 kilograms for them to survive the winter. So this is not 7 to 75 kilograms of honey, is the weight of the colony. So the colony needs to weigh at least that for them to survive the winter. Of course, you can use that as a guideline, but you may find that is slightly less for you. And that also depends if you overwinter indoors or outdoors. And some bees, some genetic lines consume more honey than others. I have noticed, and I'm sure if you have worked with different genetic lines, you might have noticed that Italian bees, they consume more honey, they eat more, and they might need more honey to survive the winter. So use that as a guideline, but you might need to adjust a little bit. And if you use single brood chamber colonies, just take a look in this book and read a little bit about the guidelines for other sizes of colonies and either indoors or outdoors. Now, how much pollen do bees need? Bees, they use pollen. So as like I said before, it's their protein source. Most of the energy that bees utilize, it's from sugars. So most of the energy that the bees are burning and using is coming from nectar, is coming from honey, but they need pollen as well. So pollen is a protein source, and mainly what they need mainly the caste that will be using lots of pollen is the workers, and nurse bees in specific will use a lot of pollen. They need a high protein diet. Bees collect pollen and they pack really tight in these cells. As you can see here, it's very, very pretty, very, very diverse. The colorful picture here is not just so it looks pretty, but also to point out that bees need a diverse food. It's very important. This pollen has they have macro and micronutrients, and these are very important for the bees. And different pollen will have different be carrying different nutrients from different plants, and bees need all of these. So they need a very diverse food, not just one single source. So bees go out, they collect pollen, bring it back, and they pack really tight in these cells, as you can see here. It's not that flower-like pollen anymore. It's nicely packed in the cells, and they also add enzymes to these pollen. And when bees bring these back to the colony, they add the enzymes, and we'll call it bee bread. But I'll talk about this in a little bit. So bees, they transport pollen back to the colony in their pollen baskets. They have hairs all over their body, as you can see in this photo. Their hairy body works really well to trap pollen and catches the pollen on the flowers when they're visiting the flower, even if they're just there to collect nectar, not specifically pollen. The pollen still get trapped and these hairs catch the pollen. So that hairy body has a function, those hairs. They have a highly modified hind leg. They have a structure in their hind leg that is called a curbicula, also known as pollen basket, like I mentioned. And that's where they will put the pollen when they are in their foraging trip. So they'll have pollen all over their body, and they can use their hind legs to brush off that pollen from their body. And when they're brushing all that pollen, they will brush to the hind leg so they can store in the hind leg on their curbicula. They also have what we call the press, the pollen press. And you can see on this third photo with a nice pollen ball on their pollen basket. It definitely doesn't look more like that flowery like pollen, right? It looks quite well packed. And that's because of the pollen press that they have on their hind leg. They brush all that pollen with also another structure that they have on their hind legs called the rake, and they can brush that, and they're using that press structure on their hind legs to sort of compact that pollen into a more nicely ball, so it doesn't fall off when they're flying back. And they transport the pollen back to the colony on their pollen basket, and they also use that same structure to transport resin, like I showed before, a bee with a resin on their pollen basket. So they will also transport resin there. Sometimes they kind of look very similar, resin ball and the pollen ball. The main difference, if you watching your bees inside of the colony and you see something that you're kind of unsure if it's a pollen or resin, The main things like the resin ball, it's kind of shiny. It's irregular shape compared to a pollen ball. Pollen balls are very nice round. The resin ball or resin load, it's very irregular shaped. So again, talking about the bee bread a little bit. So bees, like I mentioned before, when they bring the pollen to the colony, house bee will help unloading that pollen, and they will store in cells and add enzymes to that pollen. At that point, we don't call pollen anymore, we call bee bread, because again, it's something that the bees modified to be able to use it in the long term, so it doesn't go bad. So bees can store that for a long period of time, and that's something bees do very, very well. They do with nectar, and they do as well with pollen. They pack it really tight. This photo, you can see I have been sampling pollen, and we can remove pretty much the entire load of pollen from a cell just with a wooden stick, and it's very, very nicely tied together. So the bees pack that pollen pretty tight, so they can fit more in less space, basically. And what do bees use pollen for? So I kind of touched base on this a little bit. I said, and I will repeat, nurse bees require a high protein diet. So once again, pollen is their protein source, nectar carbohydrate source, and most of their energy comes from nectar, from sugars. But nurse bees require a high protein diet. And why do they require this high protein diet? Because nurse bees, they are the ones that are nursing the brood. They're feeding the brood. And although they do not feed brood directly pollen, only a very small proportion of the brood food that is actually bee bread. It's more like regurgitated bee bread, but it's only a very small proportion of the brood food that is that bee bread. Most of it, of the brood food, is a secretion that comes from two very well developed glands, hypopharyngeal gland and mandibular gland. These glands are very well developed in nurse bees, so bees that are nursing, not necessarily young bees, because like I said, they can reverse, but the bees that are nursing, they'll have these glands very well developed. And as they age, these gland kind of becomes atrophied. They're not using anymore. So when if you compare the size of these glands from a nurse bees with a forage bee, in a forage bee, they're quite tiny, and they don't use these anymore. So they're very well developed in nurse bees, and the secretion that they're using to feed the brood comes mainly from these two glands. And for bees to produce these brood food, they need a high protein diet to support the secretion from these glands. As you can see here, the very young larvae in this photo, and they're fed this jelly-like that you see, and that's the brood food. And it's kind of a whitish secretion. So again, it's not from the pollen. It's brood food. Very little. It's actually pollen. And the proportion of the brood food from each gland, either the hypopharyngeal gland or the mandibular gland, depends on the age and the caste of the brood, which tells us that the bees can discriminate. They're able to tell the age of the larvae. And for example, queens, they are fed brood food, or in this case, royal jelly. And it's mainly mandibular gland secretion. So the bees are able to tell what is the case, if it's a worker egg or if that's going to become a queen. And they are able to then adjust the amount of secretion coming from either hypopharyngeal gland or mandibular gland. Younger larvae will be fed a brood food that is mostly mandibular gland, and as that larvae ages, it switches a little bit more towards hypopharyngeal gland secretion. So that's very interesting that bees are able to tell that and are able to adjust how much of it, of that brood food that they are producing, is coming from each gland. It's pretty amazing. If you have your own bees, and I think most of you do, you also know this, of course. The honeybee colony has three cases. It's the worker, the queen, and the drone. The workers are the non-reproductive females. Queen is the only reproductive female in a colony. There's only one queen. And the drone, well, they're the male bees, and they come from unfertilized eggs. The bee's life cycle is slightly different for each case. So all of them, worker, queen, and drone, they have 3 days as an egg. So all of them have the same time as egg. The egg hatches, and then at day 4, they are larvae. So that the time is the same for the egg life stage, but is different for larvae and pupae. The drone is the one that takes longer to develop into an adult and emerge as an adult. It takes about 24 days. It can take a little bit less, a little bit more. It also depends on the genetics. It can be a little bit less or more depending on the genetic of that bee. The queen is about 16 days and the worker 21. And this is very, very important, especially for you if you are raising queens. It's very important for you to know that those life stages and how long that they are taking in each larvae state, in each life stage, because one can be a little bit more delicate than the other. And if you are raising queens, for example, there is a very important period of the bee development time that you shouldn't disturb that queen cell, or you can damage the queen while it's developing its wings, for example. So it's very important to know those times and how long they take in each life stage, and what's the total development time in general. But again, it can be a little bit more, a little bit less, and it can depend on many things. But that's just the average. They have many physiological differences, the worker, queens, and drones. And these are just a few of them, but there's a bit more physiological differences among these three. The workers, so I will kind of talk a little bit about those. So workers have wax glands and they can build comb. Queens do not have that. Workers have a barbed sting, so they sting only once and then they die. The queen has a smooth sting, so she can defend herself without dying. She's the only one. So it's very important that she does not die when she stings, although she rarely stings, but we'll talk about that in a little bit. Workers, like I said, they're non-reproductive females, so they have ovarioles, but they're not developed. So it's very, very small, and the queen is a reproductive female, so those are developed. Workers have honey crop, queens don't, and workers had food glands and queens don't. And that's just kind of a way for you to also see that queens cannot survive on their own because they cannot feed themselves and they cannot build comb. They rely 100% on workers. They cannot survive on their own. But workers also depend on the queens because they cannot reproduce. So they need a queen to lay the eggs. And drones have none of those. But drones are very important to the colony. They are the males and they are basically there for reproduction. And they can't do anything basically, but they're very important and they're very important, especially in the summer when you're trying to raise queens. You need a lot of drones to mate with your queens. When do bees raise queens? Well, there's three main reasons bees raise queens. One's before swarming, and usually your colony will be strong at that time, and that's the reason they are swarming. You will see lots of queen cells, and that usually happens in early summer when the bees are swarming. They also raise queens during supersedure. The colony can be strong or not, and they may or may not have lots of queen cells, and that can happen any time of the year. But they also raise queens during emergency situation, and that is probably when the beekeeper went there to inspect the colony and by mistake squished the queen. So the colony is queenless, and the colony has to also raise a new queen. Usually you don't see any eggs unless you cut that quite early, but usually there'll be no eggs. Can happen any time of the year, and usually again not many queen cells. Swarming is a natural behavior of colonies, and I know it can be a little bit overwhelming for the beekeeper at the beginning to try to learn everything about beekeeping and also keep ahead of a natural behavior that is swarming, because you have to keep ahead of that behavior if you want to stop it. It's a natural behavior, it's a reproductive behavior. That's how the colony reproduces. It splits in two. So it's a natural reproductive behavior. To prevent swarming, you have to split your colonies. You have to split your strong colonies, and you have to do it early in the year. Otherwise, they will swarm. So the best way to prevent that is splitting strong colonies. If you go to a colony and it has lots of queen cells, that's way too late to stop them. You're not going to stop if they have already started that swarming behavior. You just removing queen cells is not going to stop them. That's too late. You have to work ahead of them and prevent that from happening, prevent them from starting that swarming behavior. So what I like to tell beekeepers is that if you open your colony and you see lots of queen cells, instead of just trying to stop that and taking all the queen cells, just leave them, just let them swarm, because that's too late, and they will do anyways. So just it's better to just let them do it and learn from that mistake, and in the future work ahead of them and split your strong colonies, or you will lose half of your colony to your neighbor. Queens to procedure. Bees reject queens for a reason. Sometimes we do not know. Most of the times we do not know what the reason is, but there's always a reason why bees reject a queen. And when they do, when they reject a queen, they will supersede that queen, and they will raise lots of queen cells to take over the colony. And how do bees rear a queen? Well, bees, they will select very young larvae in a colony. It has to be a very young life stage, larval life stage. So they will select those very young, and I circled a few here that are in that kind of life stage, that are very young, that they can still become queens. And they will feed royal jelly to those larvae. And instead of a horizontal cell, the queen cells will be vertical. So those will become a queen cell. And the bees will raise many of them if they're superseding that queen. They'll not just create one, but they will raise many queens in the colony, many queen cells. When the queen emerges, after 5 or 7 days, after the queen emerges, she will go out on her mating flight. It's usually an average of 5 to 7 days after emerging, but that can take up to 4 weeks, and that's depend on the weather, on how if it's raining or if it's too windy. The queens will not fly. They will not fly on their mating flight, so they will wait. But if they wait more than 4 weeks, they usually just start laying drone eggs, unfertilized eggs, and they become drone layers. So it's usually in that 5 to 7 days, but it can be 4 weeks if the weather is not ideal. Mostly because drones do not fly when the weather is not perfect. So queens can also make more than one mating flight. They can make 1, 2, or 3. But they're usually very close together. It's not like they'll make one mating flight today and then next month they'll make another one. No, they will. They're very close, but they can make more than one mating flight. And they will mate with approximately 10 to 20 drones, approximately. Depends on how many drones are around. Queens do not mate with drones right next from the colonies right next to the colony she came from. She usually flies about 1.5 to 2 kilometers from the colony to mate with other drones. And that is a good thing, because that way she's also diversifying the genetics and trying to collect genetics from other locations, right? So she goes about 1.5 to 2 kilometers, and that's where she mates. And queens mate on flight. So they will fly, the drones will find her, and they will mate there on flight. When the drone mate with the queen, just after the mating occurs, the drone falls back, and when it falls back, it breaks its reproductive organ, and when it falls on the ground, it dies very shortly after falls on the ground. Half of that organ stays on the queen, and guarantees that the sperm stays inside the queen as well. The next drone that comes and finds the queen will remove that part and mate with the queen. And that happens all over, and for about 10 to 20 times. The queen stores all the sperm into their spermatheca. So they mate, they do 1, 2, or 3 mating flights, and all the sperm from those mating flights will be stored in their spermatheca. A well-mated queen will have enough eggs for her whole life, so she will not go drone layer. But if that queen did not mate well, it will not have enough sperms, and it will go drone layer. How do bees regulate temperature? Well, bees, they live, the colony lives many years. So they do experience the very high and the very low temperature, very high temperature in the summer and very low temperature in the winter. And they have very good ways to deal with that and regulate the temperature in the nest. In the winter, bees form a cluster, a very tight cluster. And here I kind of have on this picture the cluster that you usually find in the winter. Usually bees will start clustering when the temperature goes below 5 or 0 degrees. That's when the bees will start forming that cluster. And we have, we kind of, to explain how they regulate the temperature, I kind of divided in the inner circle and the outer circle of the cluster. So this inner circle that you see, we call the heater bees. So those bees will be shaking their muscles, like shivering, and they will produce heat that way. And that's what keeps that cluster warm, is by these bees that are shivering, vibrating their muscles and producing heat that way. And we also have bees that perform another job, but as important job in the cluster, that is the bees in that outer circle, and we call the insulating cluster, the insulating circle. They're kind of function as a blanket, and they keep the heat inside, but they also keep the cold outside. They kind of, so they're kind of an insulating layer. And usually they don't perform that task during the entire summer or the entire winter. They do switch when they get tired or low in energy. They will switch. The outer circle will go, some bees from the outer circle will go inside, and some bees that are inside will then go outside. All the honey and the queen, it's in the very middle of that cluster, so they can eat the honey, have energy to heat that cluster. So it's very, very important that the colony has enough honey, so they're able to keep warming that cluster. If they run out of honey, they will not be able to produce heat, and they will die. What is very common, and I see quite a bit, it's like the cluster, let's say, is way on the left, and it died. So this is in the spring. You open your colony and you see this cluster. It's a nice cluster, but it's kind of way, let's say, on the left of your colony, dead, and all the honey that it's like on that cluster, it's gone. But 2 frames to the right, there are honey, and you're like, oh, like they had honey. They just had to move a little bit, right? They just had to move 1 frame. Well, 1 frame in bee distance in the winter when it's -40, it can be quite a bit of work for them to get there. So this is just another tip when you are preparing your colonies for the winter. Just make sure they have honey throughout the colony, and there's not pockets of honey, because going from 1 frame, like 2 frames after, can be a lot of work for the bees, and they may die, and they will never know that there was honey 2 frames to the right, because it's quite of a distance for them to cover to move the whole cluster in the winter. So yeah, so just make sure that. And what about in the summer, right? And that's where the water comes, that I talked in the beginning, that water, it's very, very important for the bees in the summer, but also early spring. Bees use water to cool down the colony. So one way to kind of check this is like if you put a drop of water in your finger and you blow on it, you will notice that it gives a cool sensation in your finger compared to just blowing on a dry finger. So it cools down bees cool down the colony that way. They collect water and they spread droplets of water throughout the colonies, like on the comb, and they fan the colony to get that air in and cool down the colony. So they kind of blow air inside of the colony to cool down the colony when the air goes through those droplets of water. Of course, they don't blow. I say blow, but they use their wings and they fan to get the air to circulate throughout the colony and cool down that way. Bees also use water in early spring to dilute the honey. So if the honey is a little bit crystallized, bees can use water to dilute the honey and that way consume the honey. So once again, water is very important. I know sometimes beekeepers don't realize how important it is, but every colony needs water source nearby. So if you don't have any natural source of water, a lake or reservoir or anything, just make sure you put a bucket with water, and of course something on it that the bees can stand on it and collect the water. You can put rocks, so you can put hay or sticks, anything that can float, and so the bees can collect that water basically. Well, I guess so this was everything for today. Of course, there's much more to talk about bee biology, but I didn't want it to be too long. I think it's actually was quite long, so we can talk different aspects in more details later on. But this was kind of what I planned for today. And I wanted to ask you guys a couple of questions before we move on. So I'm just launching now. If you could answer that, that would also help me to tailor and direct these webinars a little bit more according to your beekeeping, I guess, focus. Okay, thank you. It looks like a lot of you have between 1 to 15, so that's kind of nice. But we do have a few larger size beekeepers. I do have another question. Just a second. Oh, right here. So this is just kind of a few ideas on what you would like to hear next. And if you have other suggestions, please email me topics that you're interested in, and that way will help us to choose what topics we can talk about next. Okay. So most of you said varroa and related virus, so that's good to know. But there's a lot of it's kind of spread evenly for European and pesticides. Nobody wants to hear about giant Asian hornet. Okay. And then the last thing I promise is just so we kind of have an idea on what I guess media, or what's the best way to reach out. Perfect. Thank you very much. Cass, I will quickly pass this on to you. And I'll unmute. Are you there, Cass?
Hello.
Okay. I will stop sharing my screen and then you can share yours, okay?
Sure.
Perfect.
I don't know if you want to turn on my video. You don't have to, but it might be nice for people to see.
Do I do that? Maybe. I think you have to do it on your end. Okay.
Oh. It says you cannot start your video because host has stopped it.
Try it again.
There we go. Maybe it won't. Can you see me?
Yes, I can see.
Okay. Thank you, Renata. That was really informative. I actually learned quite a bit from your presentation, and I'm going to text the B team all the stuff that I learned after this. So hello, everyone. My name is Cassandra. I work for the Honeybee Health Assurance Team, and I am an apiculture technician. So on that last survey there, it looked like 11 of you would like to learn about European foul brood. So today is your lucky day. So I'm going to give a short presentation on European foul brood, mostly because we've been seeing a higher prevalence of it in Alberta over the last couple of years, and we're just trying to increase everyone's vigilance about it. And I'll show you guys some pictures and give you some maybe management tips. But let's see if I can, just trying to figure out how to move forward. Okay, so today we're just going to talk about what European foul brood is. We're going to talk about how to identify it, and then what to do if you suspect that you have EFB in your colony, and then talk about some prevention and biosecurity management techniques as well. So I've tried to keep it under 10 minutes, but I'll do my best, and I'm sorry if this has kind of gone over time. So what is European foul brood? So it is a non-spore-forming bacterial disease. So that we usually compare EFB, European foul brood, to AFB, which is American foul brood. And American foul brood is a spore-forming bacteria, and it is essentially, well, AFB is essentially indestructible in a lot of ways, but EFB is not, it's still very contagious, but it's not indestructible, if that makes sense. So how European foul brood is spread throughout the colony, it's actually fed to the larvae by the nurse bees or left over on the cell wall from a previous bee that was infected with EFB. So not all bees that are infected with European foul brood will die, and it's actually the ones that survive that are responsible for spreading the EFB in the colony. And that's because when they're larvae and they have EFB in their digestive tract, when they go to pupate, they actually defecate in the cell, and all that infected fecal material gets deposited in the cell, and then the next bee comes over, the nurse bee or the cleaner comes and cleans up the cell, and then obviously gets infected with EFB, and that's how it spreads. So as I mentioned, the bacteria infects the digestive tract, and it actually outcompetes the larvae for food, and then unfortunately the larvae will die from starvation. So this is kind of interesting because European foul brood historically has kind of been like a spring or a stress-related disease, and it's usually brought on by a nutritional deficiency, and that can be in early spring when there isn't a lot of pollen or nectar flow. It could also be on the onset of the flow when you have the queen starts laying a whole bunch of brood, but then there are not enough nurse bees to feed the babies, and so the babies end up starving and they have a nutritional deficiency, and then the EFB can outcompete them there. But usually it would clear up in the summertime, and then we wouldn't really see signs of EFB again. So curiously, now we are seeing signs of and symptoms of EFB that have persisted into the summer, and this is really uncharacteristic. And so we've been doing some investigation into this, and this is essentially why we want everyone to be a little more vigilant. So what's changed? So there's actually 3 different strains of EFB. So we have 2 typical strains and then 1 atypical strain. This atypical strain is very contagious and very virulent. And we've just gotten results back that most of the samples that we've taken are actually of atypical ESB. And we think that is why we're seeing this uncharacteristic for our region symptoms. So here's just a picture of an AFB frame. You can see the spotty brood pattern. So we're going to move into European foul brood identification. I just want to let everybody know that I'll put this up at the end of the presentation, but on our website, we actually have a little pamphlet. It hasn't been updated with this atypical strain, but it has a lot of really good resources, and it will go through pretty much everything I'm talking about in this presentation. So definitely check that out if you guys are curious. We also have one for ASB2. So identifying ESB in your colony can be really challenging. So as I've mentioned, there's different strains, so you kind of have to consider. We actually don't, like visually, we're not able to interpret which one's which, but yes, hopefully soon. And it can affect different ages of the larvae, usually before capping. So sometimes you have larvae that are maybe like 6 days old that look infected, and there's some that maybe are closer to the capping date, and they're way more plump than the little guys. But they're a totally different color. So it can vary quite a lot. So you just kind of have to get used to what a normal larvae looks like in a normal brood pattern, and then anything that deviates from that, you want to do some investigation. And then another factor that we have to consider is that there are secondary bacteria that actually colonize the dead larvae after, so that can also change our ability to identify if it was ESB that caused the larvae death. So some of the symptoms you want to look for, we have spotty brood pattern. I mean, that is a key indication for most things, that if you're seeing a spotty brood pattern, you probably want to do a little investigation. The larvae can be discolored, and I'll show you a picture on the next slide. So it can be yellow, brown, black, red, gray. There's a variety of colors. You are likely going to be able to see the trachea, which are these little lines on the larvae, which I'll try and point out. Those may be visible. The larvae are twisted or kind of stretched in the cell, and they are not really that characteristic C shape that you usually would see in the bottom of the cell. The cell cappings may look perforated or greasy. This is probably a symptom that is less characteristic. You might find this more for AFB, because usually what happens is the larvae die before the cell is capped, so the bees will remove the larvae. But if it does, sometimes it can look perforated or greasy. If you see something that looks like ESB and you try and do the rope test where you stick the toothpick in there and you wiggle it around and then you pull it out, it should not rope more than 1.5 centimeters. And then sometimes it can also form a scale at the bottom of the cell. But then when you're curious and you're going to poke around with your toothpick, that scale actually is not adhered to the cell wall, so you can remove it, unlike AFB, which is very, very difficult to remove. So moving on to how to identify ESB in your colony. So the first thing I want to say is make sure you inspect at least 3 brood frames every time you go out, and shake all those bees off before you inspect the frame. It is so difficult to see, especially if your colony is strong, what's happening with those larvae. And so we suggest shaking all the bees off so you can get a real close look. So the first thing you want to notice is just check the brood pattern, see what it looks like. This is an example of a spotty brood pattern. So this frame is infected with ESB. And it's important to note that sometimes you might have a spotty brood pattern, but you might not see like the infected larvae in there. And of course, that's very curious. But what happens is the bees, if you have a strong colony, the bees are likely removing those larvae before you actually see them. And when you start seeing the symptoms of AFB, it's kind of like they cross a threshold, and the worker bees are no longer able to remove the larvae, like the infection's too high. So we're going to move into some pictures, and I don't know if you guys can see my mouse, maybe not, but I have some arrows drawn on here. So I'm just going to draw your attention to the top arrow here. I hope everybody can see it. But this is a frame of ESB, and you can see this top larvae is kind of curled. It's a weird color. It's like a goldish yellow color. You can kind of see the tracheal lines on there, and of course this larvae is infected with ESB. But if you move down, I guess directly below the red arrow, or the first red arrow, you can see this little white larvae here. This larvae doesn't really look like that larvae, but it's kind of twisted, and it looks like it's, I said writhing before to my colleagues, but they told me that's not a good word to use because it sounds too painful, but it's like it's moving around in its cell. That's an indication that there's something wrong, and it's also an indication of European foul brood. This other cell right here, you can see it also looks different from the first two. It's quite plump, but the color is quite different from even the one next to it. So it's a dark color. Again, you can see the tracheal lines. And then there are quite a few other ones on this frame as well. So this is a picture we took last week, and this was from a frame infected with European foul brood. And you can see there are different colors of these larvae. So it does make it a bit challenging, but sometimes you really do have to get in there with your toothpick and pull them out and see what's going on. Here is another frame. Again, we have the larvae that are kind of twisted and they look like they're kind of moving in their cell. But then next to it, you have these like darker larvae. You can see the tracheal lines are sitting in the bottom of the cell, but they're clearly very ill. This one's more yellow. So there is quite a variation, but it is, again, knowing what a healthy larvae looks like, and then anything that deviates from that, you're doing investigation. I just wanted to make a comment about these dark ones. We've been seeing a lot of these ones in the bottom of the cell. I guess it's not technically the bottom, but where that one's sitting. And it can be really hard to see them if you don't have proper lighting. I almost overlooked a whole patch of them the other day. So I was like, oh boy, they're all in there. So just making sure that you're taking the time and you're finding the right lighting, because it's important to see those. They're really dark at the bottom of the cell, and it's very hard if the comb is dark. So more, again, we have kind of like this, in this, I guess it's the left photo, kind of this deflated larvae. And then we have another kind of twisted larvae up here, and then a larvae that's kind of drying out and forming a little bit of a scale. On the right photo, again, you have, this is what I was talking about, where, you know, you have different stages that are affected. So these larvae are pretty young, and they're moving in their cell, but they're totally different than what you would see in this left photo or in the previous photos, where they're kind of a little more juicy but discolored. So getting familiar with all of these pictures is very, very important. So again, we have a larger frame here. Just, I don't know if you guys can detect it, but there's kind of like a yellow tinge to the developing larvae, and that is an indication that you might have EFB as well. And then this last photo is a close-up of EFB, and you can see those like white lines on the larvae, and those are actually the tracheal lines. So what do you do if you suspect that you have EFB? Well, the first things first is you can contact our office. So you can email b@gov.ab.ca. You can send us photos, and we can essentially see if you need an inspection. We can also send you out a sampling kit that we're trying to develop. It's in the process right now. You can also give us a call. You can send samples to the National Bee Diagnostic Center. And if you want to do that, you will have to just contact Patricia. She runs the lab there, and she can send you the sampling supply. So they use swabs. And you can also contact Renata too, and she can put you in touch or maybe help you and identify photos as well. So we kind of classified it, just so everybody knows, because this is kind of a new disease or a new strain for us, we're trying to classify the infection levels. So if you have less than 10 cells in your colony, we're saying that's like a low infection. And if they're all on one frame, we would just ask you to remove it and just burn the frame or dispose of it. A medium infection is 10 to 50 cells, and then a high infection is over 50 cells or more, and then or if more than half the brood is infected. And if you have a high infestation of EFB, give us a call and we will help you come up with a management plan. So some of the options for managing include shook swarming, and that really depends on the year. You can't really do it past July. And so we're developing a document on shook swarming, and that's essentially where you shake all your bees off and put them on brand new foundation, and then you feed them a lot. You can irradiate your equipment. You can also burn your infected frames in your colonies. And if you have quite a number of bees, I think there was a couple who had 15, you can talk to your veterinarian about getting a prescription for antibiotics, but we are trying to move away from antibiotics because they're now regulated. You used to be able to prophylactically treat with that, but now you need a prescription. So we are trying to avoid that if possible. So looking at biosecurity management practices. So I just want to hit this home because it's like super important. And I think knowing what healthy larvae and brood patterns look like is like the most important thing. So you want to see these larvae, you want to see a nice laying pattern. You want to make sure that they're white and they're kind of glossy, especially when they get a bit older, they're quite plump. Also, just to point on a side note, which I didn't realize, this drone has white eyes, and I didn't even know that was possible until today. So that's really cool. And on the right photo, that is a very, very beautiful brood pattern. This is actually a local beekeeper in Alberta. I saw his pictures and I was like, whoa, this is amazing. So this is all something we can aspire to. So just knowing that like having a tight brood pattern and healthy larvae, the white kind of glistening is something that you want to be looking for when you're inspecting your colony. And you definitely want to monitor for pests and diseases every time you go out. And because a lot of things interact synergistically, so as Renata was mentioning earlier about the mites and then transmitting the viruses, if your colony is stressed, they're going to be more susceptible to diseases. So making sure you're monitoring constantly is very beneficial for your operation. You also, for just basic biosecurity and hygienic practices, it's good to sanitize your equipment, especially your hive tool, if you're working in your operation to prevent the spread of diseases. You also want to cycle out your brood frame. So we suggest 10% per year. A lot of this is applying to a lot of the commercial people, but if you're a new beekeeper and you have all new equipment, then you probably don't have to worry for a couple of years. But if they start getting really dark and really black, you probably want to take those out of your system, because the wax does hold a lot of diseases and a lot of chemicals too. So that's not something you really want in there. And avoid buying used equipment if possible. Yeah, same kind of thing. You don't know what's in that wax. So if you're going to get a swarm or maybe a new colony, it's best to quarantine them in another yard if you can, and kind of have that set off to the side, because you want to make sure they're not diseased before you bring them into your apiary. And then you can also, to avoid EFB, you can feed your colonies during a dearth. So when there's no pollen or there's very minimal nectar and pollen around, you can feed them with pollen patties or sugar syrup. And then immediately removing symptomatic colonies or frames, etc., and disposing of them properly. It's very important. And just general like apiary sanitization and biosecurity practices include like not leaving frames out. Yeah, just making sure that, because the bees will rob if, like especially during a dearth, you might want to put an entrance reducer on there, because the bees are going to rob, which means they might be spreading diseases through robbing. So this is, hopefully I wasn't too over time, but yes, check out our website. It's just listed here. We have a whole bunch of fact sheets. Another thing I forgot to mention was we do have one on purchasing honeybees and equipment. So things you can ask. You can actually ask the seller for an inspection report before you purchase the bees, and making sure you're going through the colony yourself and asking questions like what their treatment plans were before this, all that stuff. It's all important to making sure that your colonies and your hives are healthy. But otherwise, I hope I covered it all, and I hope I'm not too over time. And thank you, everybody.
Thank you, Cass. So we have, you can raise your hand if you have a question. I didn't open for questions after my presentation, because I wanted maybe Cass to have her presentation, and then we could do questions after that. But we can use this kind of time to have questions for either Cass or myself. I did receive one question on the chat. Someone asked what was the toxic sugar that I mentioned. So like I said, nectar is mostly glucose, sucrose, and fructose. So bees eat those sugars a lot, and it's kind of their main sugar source. And there are some sugars that are toxic to bees. The one that I mentioned is galactose, and that one is toxic to bees. The other thing that is not recommended is to feed brown sugar to bees. So use brown sugar to make your sugar syrup. It is not recommended because of the amount of ash in brown sugar, and bees can't digest that. So that's not good to feed brown sugar to bees. And looks like we have another question. Someone said they're seeing quite a bit of chalk brood this year, if we have any insights. I am seeing quite a bit of chalk brood here and there as well. Usually chalk brood, it's kind of like what Cass talked about EFB at the beginning, that EFB used to be something that the bees could handle on their own, and we would see it and then it would go away on its own. Chalk brood is kind of the same thing that we see. It usually goes away, but in chalk brood is a opportunistic pathogen. So the fungus that causes chalk brood, it's thought to be kind of an opportunistic. So that's why usually the hive is able to clear on its own, but because it has this opportunistic kind of characteristic, if the hive is dealing with other things, chalk brood tends to take over because it just takes advantage of that colony that is already stressed by another pathogen, and it takes over. So without seeing your colony, if you're having chalk brood problem, I would say you're probably having another problem, and chalk brood is just taking over and using that to take over. So maybe check for other things. Maybe you have EFB or something else that it's making your colony a little bit weak, so chalk brood can take over.
I just wanted to say, oh, sorry, just one thing. Yeah, we often find like EFB, chalk brood, and sac brood. They're very common to be like kind of found on the same frame, like next to each other. So that's something as well. Sac brood is also something to look out for as well.
Yeah. Very good point. I'm sorry, I can't put my video back in. I don't know how to do it, so you have to stare my name. But if you have any questions, you can either write on the Q&A, or you can raise your hand and we can unmute you.
I think we had a raised hand.
You do, and you are the host now because I made you host, and I don't know.
Oh boy.
So you have to click on that and then unmute the person.
Okay, so I'm just gonna.
You can do
participants, panelists, attendees. Okay, so allow to talk.
There you go.
Unmute. Craig? No, did I not do it? So I've unmuted, well, hold on, I've unmuted, oh, there we go.
Perfect. Renata, you were mentioning that when you have swarm cells and the bees are wanting to swarm, you had mentioned that, yeah, just let it happen, use it as a kind of a learning experience. But with the urban beekeepers and just normal well, urban or commercial, doesn't really matter. I always think that it's best whenever we see cells in our hives, we knock them down. We'll add an extra box, and majority of the time, we actually save our hive.
Yeah, no, you're right, Craig. If it's just a few queen cells, yes. My main point, and thanks for bringing this question, just so if I wasn't that clear. What I meant is if you see lots of queen cells, if they are already in that swarming behavior and the colony has lots of queen cells, it is very difficult to pretty much impossible to stop them at that point, and they will swarm. Removing queen cells at that point when the hive has lots of queen cells, in my opinion, it's not fixing the problem. The problem is that they ran out of space and they are already in that swarming behavior. So in the future, you have to kind of work ahead of that and give them more space. But if they do have just a few queen cells, yes, by all means, like remove them, because if you just have a few queen cells, and bees raise queen cells all the time. So if you just have a few queen cells, remove them, but also take that as a just as a nudge that the bees are telling you, hey, maybe I need some space. So maybe take a step back and think about, do they need more space? Should I put another box? Some genetics are very like into swarming, and again, that can be genetics as well. So if you have these genetics that the bees are very swarming like, they will swarm no matter what you put. Like if you put a lot of space, a lot of boxes, they will still swarm, and maybe that's not the bees you want. Maybe you want a different genetics, but if you just have a few queen cells, yes, remove them.
Sounds good.
Awesome. Thanks.
Is it?
Do you want me to put you back on as?
Oh, as host.
There you go. Oh, yes.
Oh, there you go.
Now, there you go.
Someone is asking, have you guys researched and or used super DFM Honeybee for gut health, for helping with nosema? I'm sorry, what does, do you know DFM, what that stands for, Cass?
I don't, but I've heard it before, but I have not.
Super. So to the person that asked the question, can you kind of explain what super DFM means? I'm assuming. I mean, I have heard a lot on bee health. There's some supplements that are out there. Okay, by strong microbes, probiotics. Okay, yeah, that's what I thought. Yeah, those. There is, I have not researched that. I don't know if in the CDC you guys have done anything with probiotics, no. But there is a researcher from University of Saskatchewan, the veterinary school, and his name just, I cannot remember, but he studies honeybees, and he did a lot of work on eczema and with probiotics, and he presented some of his preliminary results last year during our convention in November, and he didn't find that that these probiotics did anything to the nosema levels. The only treatment that had an effect on the nosema levels to go down and control nosema levels was fumigillin. And he studied quite a few probiotics, I think 3. And you can probably go into his website and check which elements.
Oh, Elamir Simcoe.
Yes. Elamir Simcoe from University of Saskatchewan, and he did that, and he did not find that probiotics helped with nosema. So only fumigillin. What I have heard though about these supplements is that sometimes maybe whatever they're selling that supplement for, oh, it's going to help you with this or with that, maybe it's not going to. But what I have heard from beekeepers is that these some of these supplements, they just make the syrup more attractive to bees, and bees just drink the whole syrup. So if it may not be beneficial to treat whatever he's saying that it is doing, but if your bees are not taking the feed and adding that makes them take the feed, then that's good. But it's kind of a slippery. You have to kind of test it and see for your own. Go ahead, Cass.
Well, I was just going to say too, I feel like there isn't really like bee-specific probiotics as well. Like a lot of the probiotics are for other animals. And so it's kind of inconclusive, as you were saying, whether it's beneficial or not. But I'm not sure if there's any research that's been done about whether it's harmful.
Yeah, that's true. And I'm not sure if there is any research saying it's harmful. And there may not be a huge benefit either. So maybe, what do you say in English, snake oil? Is that correct? Snake oil? Is that an expression that you say when you buy something?
Yeah.
Okay. So it might be that. But as long as it's not bad for your bees and you want to try it and see how it works for yourself, then that's good. Okay, I don't think we have any more questions. And if I missed your question, I'm so sorry. But just want to thank you, Cass, for logging in and giving this presentation. It's very important to have more information on EFB, especially this year that we have had so much. And thank you all for signing in, for joining us on our first Hive-side Chat. And we'll be in touch for our next one, the next topic. And I'll send emails with a date and a time again. Thank you very much, everybody, and have a good night. Thank you, Cass.
Thanks, Renata, for inviting me.